All right, so we're looking at the regulation of gene expression in prokaryotic cells. So really what we're looking at here is operons. So quick overview, we'll look at what the operon structure is, followed by some examples using tryptophan and the lac operon for lactose. Two different things control the lac operon, lactose and glucose, so we'll look at those separately. To be able to understand operons, we need to really go through negative feedback and make sure you're solid there. So essentially what we're doing is trying to keep everything at a certain rate or a certain level. In this case, we're looking at chemicals, but we can also look at it from the point of view of like a temperature or anything like that. When it's too high, you have to do something to bring it back down. When it's too low, you have to do something to bring it back up. So you're constantly like honing in right around a set point. In the case of our operons, we're really looking at tryptophan and lactose, but that can vary, and wanting to make sure we just have certain amounts of certain products. So okay, that's what negative feedback is. It's just negating whatever brought you out of homeostasis. And so prokaryotes are going to use this type of system in order to have enough energy and have the resources that they need to build proteins. Now, just in general, for any kind of negative feedback, there's two ways that we can look at from a product standpoint. We can look at it from the point of view of end product inhibition right here, which is a fancy way of saying that whatever you've made at the end of a metabolic pathway then negates the earlier enzyme so that you shut the pathway down. Once you don't have enough of it anymore, that value raises back up. On the gene expression side over here, you can actually have the gene product. For example, let's say that we've got A, B, C, and D here. If we've got these various enzymes that are converting A to B, B to C, C to D, then whatever this product here is can actually inhibit the gene for the enzyme from being produced. If there's no more enzyme here, then A isn't converted to B, and therefore D isn't made, and you save your resources early on, like, for example, your reactant A, and you don't make too much of whatever is at the end. I hope that made sense. Okay, typically anabolic pathways, so remember that's anything that is building something up, that's often turned off by end products. I'll underline it, turned off by end products, whereas catabolic pathways, which are those that cause the breakdown of substances, are often turned on by the reactant, and we'll look at why that happens. Okay, so we can now start looking at this in terms of the structure of operons. You've got to make sure that you understand this structure piece before moving too far forward, because we're going to refer a lot to things like promoters and operators and so on. Okay, so two main areas that we're looking at in terms of genes that are getting made. So remember, genes code for mRNA, which codes for proteins. So essentially, a gene makes a protein after a couple of steps. So we'll be looking at structural genes that are part of the operon. So for example, these can be enzymes or transporters, really any kind of protein, and we know all the functions of proteins now. So any of those that are closely related to doing a certain task are going to be the structural genes that are under control of one operon. And then the regulatory gene is outside of the operon, and this is going to make some kind of regulatory product. We'll talk about that too here in a little bit. Regulatory product. Okay. All right. Now we also have two kind of switches right here, the promoter and the operator. The promoter is going to be where the RNA polymerase binds, and the operator is kind of like a block, a way to block the RNA polymerase from being able to move forward. So this in effect acts as an off switch. Okay. So the regulatory genes I've covered. Promoter, operator, structural genes. Yay, good, I have everything here. Okay. Furthermore, we can talk about two types of gene regulation, either negative or positive. Anytime we use a repressor, then we're going to be thinking of negative gene regulation. Anytime we use an activator, we're thinking positive. Repressors are going to be anything that stops gene regulation from happening, and it's going to do so by binding to that operator. So remember, this is the off switch, which means if a repressor is active, then it's going to turn the system off. Conversely, if it's inactive, then it's going to act to turn the off switch off, effectively turning it on. Okay? In other words, repressors can constitutively be active or inactive and be turned on or off. Anytime you turn on a repressor, that means you're going to have no transcription. If its repressor is off, you're going to get transcription, essentially inducing transcription to happen by turning the repressor off. On the flip side, positive gene regulation just means that we have some kind of activator that is binding to the promoter and allowing transcription to happen at a better rate. Let's summarize all of this right here. Okay, so regulatory genes are either going to make repressors or activators. We don't really look at inducers in terms of regulation, so don't worry too much there. The promoter is where we've got the polymerase binding, and activators are going to bind here to increase it. On the flip side, operators can be blocked. If you block an operator with a repressor, that means that transcription doesn't happen. Okay, let's start with the trip operon, TRP for, oops, there we go, tryptophan. Tryptophan is an amino acid, and so if you don't have tryptophan available in your environment, if you're a single-celled organism, you need to make it. Otherwise, you can't make proteins. So what we're going to have here is a regulatory gene product that is an inactive repressor. It's going to be this guy right here, okay? This is inactive. It is not repressing because it's just chilling. And so long as you don't have tryptophan, this remains inactive. So that means without an active repressor, the operator is on and transcription occurs, okay? So that's this guy in the absence of tryptophan. All right, that makes sense. If you don't have tryptophan, you need to make it. And these are the enzymes for making tryptophan. On the converse, if you do have tryptophan, there's no reason to make it. So you want to turn this operator off, which means you want to activate the repressor. And so here's our inactive repressor right here. When they bind together, you have an active repressor. And for that reason, we're going to consider tryptophan as a co-repressor. It works with the repressor to repress the action of the operon, okay? So active repressor means that this gene is effectively turned off, and the enzymes for making tryptophan are no longer expressed because you don't need them. You already have tryptophan. Why make it? As soon as tryptophan levels fall back down to a certain threshold, then this system kind of breaks down. The tryptophan breaks away from the repressor goes back to being inactive, and voila, you're back to this scenario. Okay, now when we're looking at the lac operon, this is a separate operon altogether, and it is based on lactose. So, the gene products here include beta-galactosidase. And beta-galactosidase is an enzyme that is going to convert lactose into glucose. So the whole purpose here is to be able to make energy. Glucose is the first step in being able to make ATP. So if I need energy, then it's great to be able to use lactose that's in my environment. Okay? So the regulatory gene product in this case is an active repressor. Last time we saw an inactive one. This one is active when nothing is bound to it. Okay? If nothing is bound to it, that means there's no lactose. That shuts the system down. In other words, don't make the enzyme to break down lactose when there's not even lactose in the first place. And in fact, it's not going to be lactose that's binding there. I feel like pink is a good color. Instead, it's going to be allolactose, which is an isomer. So allolactose is in equilibrium with lactose. When you have one, you have both. Okay, but if you do have lactose in your environment, it's important to be able to then break it down, because lactose is a disaccharide, and you want to be able to make glucose from it, which is a monosaccharide that you can then use to make ATP, right? And so that's the beta-galactosidase. So if you have the disaccharide, you have to be able to break it down into the monosaccharide. Otherwise, it's pointless. That's where beta-galactosidase comes in. That's where this operon comes in. So when allolactose is present, it's going to bind to the active repressor, and that's going to inactivate it. That means that this can no longer bind to the operator, and therefore the operator remains on. Okay, so what are we going to call that? Because we have now induced transcription to happen, this is called an inducer. Okay, allolactose is the inducer because it induces the expression of the genes for beta-galactosidase and other protein products that you don't need to worry about. Okay, so to kind of summarize this, if you have lactose, then you'll start making the enzymes to break down lactose. If you don't have lactose, it doesn't make sense to make the enzymes because you don't have it anyway. Okay, there's one other little thing we need to include here to really understand what's going on. So add a page below. Let's just make a plain piece of paper. Okay, you need to understand that adenine and 5-carbon sugar and phosphate and phosphate and phosphate. So this is my energy molecule, ATP. When I have a fully charged happy cell, it is full of ATP. And I get that energy, I make my ATP by breaking down glucose. So every time I use my ATP, it gets coupled with another reaction. Then instead of ATP, I'm going to end up with ADP, right? That means that I have lost a phosphate group. I have gone down in energy. Well, turns out we can go even lower than that, right? I can make ADP, adenosine diphosphate, now into AMP, adenosine monophosphate. That's like a really, really, really dead battery. Let's pop this little guy in here so you know, like, there's nothing left in here, right? ATP is beautifully charged. AMP is like kind of weak, but it can at least couple some reactions. But AMP, that's hella dead. And if you have cells that are full of AMP, that means you have very low levels of energy. And if you have very low levels of energy, what that means is that you must have low levels of glucose. Okay? Think that one through for a second. So if you don't have much glucose, you're going to have high levels of AMP. So low glucose equals high AMP. Now you'll notice I put a little C there. That just means cyclic, which is a fancy way of saying that something binds an extra time. It's actually over here. There we go. Doesn't matter. Either way, you have a high amount of this very low energy molecule because you have, a high amount of the low energy molecule because you have a low amount of glucose. And conversely, when you have plenty of glucose, you're going to have plenty of ATP and low AMP. Okay, why does this matter? Well, because in addition to lactose, the lac operon is also going to be controlled by glucose. Oh, it's the wrong button. Let's try that again. Okay, glucose. And why would that be? Well, if you have glucose, the cell is going to prefer to use glucose before making the stuff to break down lactose just to be able to make glucose anyway. So if the cell already has glucose, then it's going to kind of shut down the lac operon also. Whereas if the cell does not have glucose, then it's going to activate the system. Okay, so here we go. Now our regulatory gene product is going to be an activator. An activator is something that binds to promoter. This is new. I don't know if that's ER or OR, doesn't really matter. But it's binding to the promoter, not the operator. Everything up to this point has been binding over here to the operator. This time we're going on to the promoter. And what the purpose of an activator is to allow RNA polymerase to bind really, really well. And if RNA polymerase can bind really well to the promoter, then transcription can happen, because remember, RNA polymerase is going to be what reads the DNA to be able to make mRNA. So there's our regulatory gene product, is an activator called CAP. Here's CAP. And CAP is in an inactive form. So right here, we can see CAP is inactive. It's just chilling. Both cases here, by the way, we're imagining we have lactose present. This right here is representative of positive lac or presence of lactose. Okay, so that is to say we're not blocking the lac operon from working. Okay, back to this right here. We've got CAP, but it's inactive. So right now, it is not increasing the presence, the binding of the RNA polymerase here. The only time CAP is going to become activated is when it binds to cyclic AMP. Cyclic AMP is present when there is a very low amount of energy. In other words, low energy means that we don't have much glucose. So if we've got low glucose, then we have low energy, which means we have high cyclic AMP, which means because of the fact that we're low on glucose, and I say we like I'm a bacteria, which at this point you're probably looking at me like I am, but that's okay. Anyway, so we have low glucose, so we need to use something. And if glucose isn't available, we're going to go with our second best, which is lactose. So if lactose is available, let's go ahead and utilize it. This is our plan B. That's not meant to look like a beta. That's our plan B, since we can't make glucose work because there is none, because if there was glucose, it would have been made into ATP already, and I wouldn't have camp in the first place. So since there is not glucose present, hello, we're going to have to switch over to using lactose, which means upregulate the amount of beta-galactosidase so that we can make glucose from